Welcome you all for my last part of my talk. I will try to summarize, and as I have told you in the starting of my talk, that I will take you to some of the example from my own lab, from done by our different collaborator with my collaborator, which our lab have done. So let's start with the recent one first, and then I'll take you to what we have done besides this new one. And everybody is now, talking about the COVID. Everybody is know about it, and so I'll just take you to that component first. Okay, so recently, even in the last week only, I could say, this work has been published by our group. This is a very good example of, the collaborative research. And this work has been done by 7 organizations, not only from India, even abroad. And 3 organizations from India, Jamia, Deshbandhu College, Hansraj College, and besides there's one of the American university and one of the Saudi Arabia university. So almost the 7 organizations involved for this work, and we got very good appreciation from the editor-in-chief as well. So let's start with what we have done in this work. Everybody is aware about the COVID-19 nowadays, especially the other way you can say SARS-CoV-2. The recent exposure to SARS-CoV-2 has affected entire, the world, and resulted more than, rather it was the, you can say that 1 month old data, so 0.3 million deaths. And SARS-CoV-2, despite sharing 80% of the genome similarity to the SARS-CoV, talking about the previous, the strain, its potential drug targets are highly conserved. Despite high conservation among the target proteins, the structural difference make available drug ineffective against SARS-CoV-2. The reason is the cytokine storms associated with viral inflammatory responses, as seen in case of SARS-CoV-2 infection, which may be targeted to handle COVID-19 patients. Experimental trials of the various FDA-approved drugs are showing positive effect. But further evaluation of the large subject groups is required to confirm their efficacy. So before I'll take you to the detail about my work, I will just would like to take you for first about the what is the life cycle of this, how it effect basically, how this SARS-CoV-2 enter to the host cell, how they complete their life cycle in our host. So maybe many of you must be knowing, if whosoever working in biology, but I came to know that many of you are not from biological or direct research related to the virology. So I just give you simple glance the life cycle of the SARS-CoV-2, showing, if you see, start from here, entry point from here, receptors, they bind, and then you just follow me like this way, Okay, so the life cycle of SARS-CoV-2 showing potential drug target in the host. The S protein, you can see here, of the virus binds to the cellular receptor that is called ACE2, followed by entry of the viral RNA genome into the host cell. After the genome entry into the host, into the cell, translation of a structure and NSPs follows. ORF1A and ORF1AB are translated to produce polyproteins PP1A and PP1AB, which are further cleaved by proteases that are encoded by ORF1A to yield 16 non-structural proteins NSP1 to 1sp60. And assembly and budding into the lumen of the ERGIC, that is endoplasmic reticulum Golgi, in the intermediate compartment, then follows, like here you can see, right? And variants are finally released from infected cells through infected cells through exocytosis. And in this life cycle, coronavirus multiple stages are being seen as potential druggable targets. And drugs working like S protein inhibitors, RNA-dependent RNA polymerase inhibitors like remdesivir, flavipivir, galidesivir, ribavirin, protease inhibitor like lopinavir, like ritonavir inhibitors, like federatinib, like barbicitinib, many of them, like monoclonal antibodies like tocilizumab, so have been proposed to show promising effect against the novel virus. And taking cas-based approaches from previously encountered viruses like SARS and MERS, many drugs are facing clinical trials, and I hope very soon we'll have the drugs for this SARS-CoV-2 in the market. The genomic feature, just because I have done the in silico components, I just try to conclude our our conclusion from our group research. Like the genomic features of SARS-CoV-2 discussed in this study is basically a possible hypothesis of pathogenesis and transmission in the human disease. And efforts in the short term should be focused on the developing a vaccine or the inhibitor by the targeting the major viral proteins such as S, E, M, and proteins, or RDRPM proteases. The three infections have marginal differences in terms of innate immune responses, but greatly differs in terms of morbidity and mortality. The details insights presented that the novel coronavirus differs in its modus operandi compared to the previous known strain, that is SARS or MERS. Our comparative study provides answer in the context of developing potent drug and vaccines against protein targets for development of better approach in COVID-19 therapy. Second work, just 2 years before, we did as the targeting receptor tyrosine kinase like orphan receptor ROR1 for cancer treatment using in silico drug discovery approach. And here, we have targeted ROR1 because this is a member of RTK family and is primarily expressed during the early stage of embryogenesis. And it has been reported that potential target for diagnosis and development of therapies against a variety of different human cancer. And we draw conclusion, and finally a molecule is named as beta 1, 2, 3, 4, 6 penta-O-gliol D-glucopyranose. And this compound belongs to the class organic compound known as tannine. So I hope, and I'm still working on it for the organoid system trial with the help of University of Southern California at KK School of Medicine. Third part, just again, already done in our laboratory, and where we have, to find the structure and function insight into the tag accumulating hydrolyzed protein of the mycobacterium. And, that also give very good results that, structure analysis of this suggests that hydrolase-like activity and probable involvement in the host lipid import and the tag accumulation, which plays a very important role, especially when you talk about the MTB drug resistance. I told you that besides the human health, I will take you a little bit about the agriculture side also. And there also, last year only, we have designed a tool called NeuroPyPred, which is a computational tool to predict, design, and scan insect neuropeptides. We are still working on human neuropeptides, but this is very, one of the important and most versatile group of neurotransmitter, or we can say neuromodulator, and secreted by the central nervous system, which regulates various behavior and physiological activities, and which have small peptides around 5 to 80 amino acids. Neuropeptides are basically omnipresent in living organism. Different group according to their function includes, myotropins, diuretic, AKH, RPCH family, eclosion hormones, pheromones, biosynthesis activity peptides, allotropins, allostatins, ecdiosteroidesis, ostatic hormones, almost present, omnipresent in almost all organisms. And these insect neuropeptides and their associated receptors have been one of the potential target for the pest control. So this is basically we already released. Now it is available for use for the public domain, and you can just type Google neuropiperate, and you will reach to the home page, and you can facilities like you can predict, you can design peptide, you can do the protein scanning, you can do the blast, you can download all the modules for our database and this study. So I request, if any of you are interested, either you can write me or you can visit and use these tools. Another very interesting work we perform is pathogenesis related protein and defensin for the plants, but again, allergen for the human. Pathogenesis related PR, what is PR protein? Are basically defensin expressed upon biotic and abiotic stress by the plants, and they are also synthesized in specific plants parts such as roots, stems, leaves, pollen, grains, and fruits. And according to the current classification, there are 19 different classes of PR proteins. 8 out of 19 are proven to exhibit allergic reaction in human. Here we carried out the allergenicity potential test for the in silico for the remaining 11 classes of PR proteins using in silico approaches here, and you can see all the diagram, which is very self-explanatory. Here you can see the how allergens and what are the how the allergic reactions will do, hypertension, conjunctivitis, rhinitis. What I'm talking about is PR protein, So how they are create like skin rashes, they may cause asthma, they may cause, oral allergy. Yeah. So and sometimes they may cause, nausea as well. So this proteins is, good for the it is defensin for the insect and allergen for the human. So the major work what we conclude in our study is like if you see the model the protein and for which and I just repeat in a way other form another way, like allergenicity is basically the major cause of concern as a huge huge proportion of the population suffers from one or other allergenic disease for which many times the cause remains undetected. Hence, it is important to understand about the presence of allergens in the food products and that are consumed on everyday basis. Our in silico analysis of different families of PR proteins suggests that these proteins can trigger allergic response and can even trigger a cross-reactivity response. Henceforth, we strongly support that some prior knowledge about the B cell epitope binding residues and interaction between the allergens and antibodies could provide important insight to understand various allergic responses invoked by different families of PR proteins, and this can help clinician to provide a tailor-made dose of various drug available to treat different symptoms triggered by different allergens. We also conclude that because the proteins are dynamic in nature, therefore it will be interesting to see how they interact with other antibodies molecules and how does the different forces operating during the protein-protein interaction, stabilizing them to attain a particular orientation with the most stabilizing interactive energy. Okay, so recently another work we did the molecular modeling of the chemosensory protein 3 for Spirulina and its binding property with the plant defensive metabolites. This work also very important in the sense, to design CSP based, advanced biosensor to control, insect pest management. And another project besides this work, which I have shown you just last few minutes, another work which is going on in my lab is related to agri-informatics, cancer, tuberculosis, and rare genetic disorders like FOP, fibrodysplasia ossificans progressiva. And this disease is basically a rare genetic disease. Recently, I got a project from ICMR. And although in India it is not, this is rare genetic disorder, so hardly we have approximately, this is the statistical probability that almost 660 cases of FOP may be in India. And this is basically an inheritance is autosomal dominant, and although most cases are due to the sporadic new mutations. Another work which is going on is lead generation of the novel inhibitors. For AKRC1 family, one member for the treatment of the breast, cervical, and endometrial cancer. Another work is going on designing the novel inhibitor for MCL1 for the treatment of the human cancer using pharmacophore modeling approach, the approach which I have already shown you in like CAD model and all. And now I will take you to why, if you are from computer science, if you are from mathematical science, if you are from biological science, collaborate all of you and design your own database and web tools. Like there is 5 categories of database here, like Encyclopedia of Cancer Genomics data, tools data analysis of interaction, like CAS, JAD, and tool used for visualization, database focusing on association of the specific biological features to the cancer, and database for drug discovery. So I'm just giving you some examples. You can just note down the name and you can visit the sites, and these all databases are freely available. If you have institutional ID, you can use these all databases and tools. You say NAG binder, you can go for the NAG binder, then cancer SSP, you can go for the PCMDB, you can go for the ApoCandy, you can go for the other software tools which I have told you. And it is, not that difficult. Only thing is, if you have a good concept of biology, you collaborate with a person who is from, computer science, if a person from mathematical modeling, and, come together and you can have very good. So another important aspect is like Entrepreneurship Campus. Entrepreneurship Campus empower the social entrepreneurs, especially young people from around the world, to create a culture for entrepreneurship, to promote entrepreneurial education and engage in more peaceful work. Participants are invited to submit their innovative ideas, projects with societal impact, which champions and implement one or more of the sustainable development goals. For example, I told you some of the Indian startup is like Ganit Lab, which is a Bangalore-based biotech startup lab, which is working on the genome science and bioinformatics and computational biology tools to solve scientific problems. It is founded by Vinay Panda in 2011. You can go for Mars Design, you can go for the virtual veterinarian, and you can go for Docttocare, and this is a leading healthcare online portal that enable user for non-metro cities access the book surgery appointments with doctors and hospitals. You can go for Cultivate. Is also set to revolutionize the agriculture practices with its automated farming system built on artificial intelligence and the Internet of Things, IoT people in short say, and the farming solution aims to deliver a high quality crop output. And then you may go for Vitor Health Sciences Privately Limited, a combination of vitality plus vigor. You can go for Wanted Note, a fintech ecosystem for pharmacies combining finances and fulfillment. You can go for the Kisan Gate, which is a young and innovative startup with the motive of revolutionize the agro sector through digital platforms that are synergistic with the environment and business needs. It sees the problem persist in agricultural value chain and the solution effort to do take user-centric approach, leaving most of the solution as a raw or over-engineered methods that are difficult for easy adoption and scale up. So conclusion, I should say after all this discussion, I try to cover as much as possible, but within 1 hour is difficult. Although the main conclusion I should draw from translational research is policies and support mechanism that promote translational research need to be put in place. Clear definition and awareness of patenting, intellectual property right, and research, exemptions. Viewing translational research as a circular or iterative process. Consideration of the collaboration as a key ingredient for successful entrepreneurship in translational research. It's my personal experience. If you collaborate with different group of scientists, different group of worker, different group of researcher, industry person, then only you will come out with some products. These are the references which I have used for my, the presentation. These are the few recent articles which I have already discussed with you, the details. Neuropiperate, like recently I told you, molecular modeling of CSP, and this is the recent work just last year only. You may visit, it is available online now, the work on the SARS-CoV-2, and I must thank, the collaborators from Germany, from US, from India, like Professor DPS Raghava, Professor Henge, Professor Ayan Baldwin, Pali, Professor Verma, Dr. Singh, and Imtiaz Hassan. And these are, my lab tree, because without whom I can't run my lab. So thank you so much. They are the real stakeholder, and these are my lab members. And last, I could say all of us do not have the equal talent, but yes, all of us have an equal opportunity to develop our talent. This is said by our former president, great APJ Abdul Kalam ji. So, we have to do a lot of things, a lot of good work for our country to, come out with solution of a lot of ongoing problems related to health. I always focus, health is important, food is important. So I have focused here with the, health science and agriculture science. Thank you so much. And I must thank organizer team of TLC Ramanujan College to giving me opportunity to interact with you. Thank you all. Good luck. Stay safe. Stay home. Thank you.